So, Jack Szostak, Elizabeth Blackburn, Carol Greider, welcome to Stockholm.
Thank you.
Pleasure to be here.
And your Nobel Prize is, of course, associated with a couple of interesting numbers. This is the 100th time, as I'm sure you've heard, that the Nobel Prize in Physiology or Medicine has been awarded. And also this is the first time ever that two women have been co-recipients of the same Nobel Prize in the sciences. And telomere research in general is one where the proportion of women is more normal, if you like. It's more representative of the makeup of society. Is there a reason for that, do you think?
I think we should turn it around and ask why everybody else is so aberrant. Seriously, because I think that actually might frame the question a little bit more instructively as to why things are not, as you said, the biological or societal representation of women. And we can point to various sorts of things, such as the fact that some of us who've been in the field are females, but actually there are plenty of men in the field as well. And I think what stands out is that the numbers are a little different. But certainly having examples of women who've done well in science, we all know that kind of example does make it more encouraging for others, younger people, to visualize themselves being successful in science. And so I think there's been a kind of perpetuating effect there. But I do like to think that this is the normal way it can be, and perhaps we should think about, well, how do we make this more normal in other fields of science?
Well, picking up your question, why is it aberrant in other fields, do you think?
Somebody else can take that one.
No, there's obviously a lot of historical reasons and bias from early days that takes a long time to get rid of, and a lot of good role models to get rid of.
Have you found it difficult being a woman in science?
No, I haven't found it difficult, but I think that one of the things that I've always done is sort of just put blinders on and have done what I wanted to do. But it was when I then started to get to the higher levels in ranks that I could then look back and see what the data was. Graduate school, there was 50% women in graduate school. As postdoctoral fellows, usually 50%. But then, as you get higher and higher up, the representation is lower. And while I didn't feel like I personally had experienced any big obstacles, I'm a scientist. I can look at the data and see that there's something that's not quite representative as one moves up the higher ranks. And so I think role models are one good thing, that the more people there are in the higher ranks, the more the younger people can look up and say, oh yes, it's something that I can do, and it doesn't seem like an impossible task.
It's one thing. Is there more that needs to be done than role models? I mean, would it naturally change, or does it have to be aggressively tackled, do you think?
I think aggressive tackling is good. I mean, look at something like smoking, right? It took very aggressive actions to make smoking less of a widespread practice and a certain amount of imposition of things. I don't think it'll happen organically, completely from within, although my colleagues, when you talk to colleagues, they're individually very well disposed to the idea that they see the value of having more women and more diverse groups of people in science, because like any enterprise, the more diverse sorts of backgrounds come into it, the better the way the problems can be solved. And so I think there's not a lack of goodwill among a lot of people to do this. It's a question of how do you do it, and I'm sure a mixture of various sorts of strategies will be helpful. Not least education, and not least just seeing a woman there sort of makes it more possible. I think we're a very visual species, and you look at something and that's the evidence in front of you that you seem to think, oh, that's me. If you were a young woman, I could do this. I think, though, that we have to make the career structure a little bit more flexible, because there's been a one-size-fits-all model for careers in science, which have been very much that based on, a man having a supportive wife or partner or something to sort of take care of life and family, and that's been something that is daunting, we find, to young women. I personally found it very daunting as a young woman to enter the career structure, not the science, but
Yes.
the career structure. And so I think we could be much more imaginative about how we make sure women don't leave sciences during the time when they have preoccupations with family or with elderly, parents who need long-term care, and those sorts of things that women often do. So.
Yeah, that requires one to have really quite a strong support structure within science, which presumably
That's right. And so there has to be
brought more broadly than that. And there are a lot of things done here in Sweden that would be kind of shocking in
Yes.
the United States.
Yes,
right?
yes,
More
absolutely.
broad support in terms of general family
Yeah.
support.
Yes, yes. Because I think the sticking point for many women, at least in my personal experience from what I see, is they say, I just can't see being a scientist, and I also do want to have a life. And I think that's something that we shouldn't say to young people, oh no, you can't have a life and be a scientist. And yet that's the perception that they have. And I think as senior scientists, we could do a lot more to try to think of active ways in which this one kind of career structure model that we have could be thought of more imaginatively. And I just know of examples of women who've gone part-time as their families and other needs have happened, and then they go back in full-time and they just come roaring back, and have done really well. And they're not all in the United States, unfortunately, which again tells us something about the United States situation and support.
Mm-hmm. So turning to telomeres, the blobs on the end of chromosomes, as I think that's you're using it with your own phrases. I've heard you say.
Probably.
They were known. They were known in the 30s, and it was known that they had important protective roles for chromosomes, but people didn't know what they were. And it was you in the late 70s that sorted out their molecular nature.
Began to. It's still an ongoing saga. None of us are out of jobs yet. That's right. So I think it was so wonderful, the science that was done cytogenetically, in which such deductions were made about what's going on with chromosomes and their inheritance. And now we look back at it and we say, they didn't even know it was DNA. And yet the thinking about what was observed, and I particularly enjoy McClintock's work because that's what I got more familiar with, was so elegant, and there's so many treasures in there of insights into what turns out to be going on in meiosis, and as it turned out, with what's going on with telomeres, although her fame more broadly was, I think, perhaps more for jumping genes for many years. What she said in something written in 1931 about a telomere being distinctive was, and she didn't call it a telomere, was so clear and I think very important. And it was just lack of the research tools of the time. The research tools of the time. Right. They had microscopes, they had genes, they had phenotypes, right? I mean, they had really wonderful other kinds of thinking, but they weren't playing with the same sort of toys in the lab that we play with. That's right. Yes.
And then you two met at this Gordon conference in 1980, and it's sort of the way science is supposed to happen. Two people meet each other, have an idea, and do an experiment, and it proves something wonderful.
Yes. And NIH, nobody pays for it. But they do pay for it. No, they pay for a broad sort of setting in which you can go and do experiments and explore and ask questions without somebody saying, oh, is this going to be useful for this year's economy or for, somebody or other's medicine? I think that's really important, right? I mean, we really thought that was something that we didn't even think about at the time. It was such a given that you would go and do an experiment with broad funding that, is never going to be wasted. Scientists don't throw away money. They work very hard, and giving scientists money to ask questions that, hadn't been planned. It's really important.
That meeting, or that kind of meeting, it's great for bringing people together, who are working on different things, and you know, so
Bond over horrible food. You know, right? These meetings are famous for having bad food.
You hadn't
That's
met
right.
before, though, right? The
No.
two of you had met. You had met. You had
that's
never met
when we
before.
met. After
No,
your
you
talk,
walked up, we walked
I
and
came
we started.
up.
We walked across this lawn and just talked and talked. Yeah, it was very cool.
So I was working on broken DNA molecules, ends, and then Liz had these ends that behaved completely differently. And it was just the contrast was kind of shocking. So we had to talk about it and, you know, see what we could do to figure out what was going on.
So Liz, you gave your talk first,
Yes, yes.
and then okay.
And so it was after the talk that the two of you teamed
Yes,
up and
because
saw
I
that there was a
talked about what we knew about the molecular nature of these DNA molecules. You know, the ends you could get your hands physically on, and
We turn to the experiment in a minute, but I just wanted to ask you in general about choosing companions in science, because it's so important to get the right people to work with. Is there some, and of course one works with lots of different people through one's career, but are there some criteria that you apply to choosing companions to work with?
I think it's mostly a matter of, you know, is there an interesting experiment that actually can get done?
yes. Yes, and then if you have the luxury of choice, it's yes, you do want to work with people who really do rigorous science, I think, and that can be very different kinds of science can be done with rigor, but I think that's important as one collaborates with people outside your area. And Jack and I, we were really much in the same sort of area, right? It was a molecular genetic tradition. But when you collaborate even further out, where now you don't have the deep expertise, then I think you have to have a real respect for that person's quality of their research.
You also need to have
yes.
someone, you know, who's fun to talk to and you can exchange
Yes.
ideas with easily. And, you know, some people are better at that than others.
But you're right.
More fun to work with.
But you're right that if the science is really exciting, you'd make a way.
You'd find a way.
Yeah, yeah. But
And when you're picking companions who don't have a track record, students to come into the lab, what do you look for in them? Is there...
Really, it's just the excitement about what they're doing and this ability to communicate that excitement back and forth. And I mean, I think if people really are interested and care, that in just interacting with them over a period of a few weeks, one can tell whether or not there's a compatible set of interests that are there. So usually there's an opportunity to do that when students may be coming into the lab. You have a chance to get to know them a little bit and see where the compatibilities lie on both sides. It's not just a mentor choosing a student there's opportunities for students to go around and choose mentors and make sure that they're compatible with them as well. And I think that both of those things are important.
Well, I was going to go on to ask, what do you think are important? What do you think are the important characteristics of being a mentor? What do you try and provide for the student?
Again, it's all about the science, and it's about being able to sit down and have a conversation and really understand that person's interest. Some problems are very interesting, but somebody may approach it from a particular angle and somebody else approach it from a different angle. And then when those two people talk, it may not be as easy to understand, but a third angle, there may be, shared understanding, like, languages. If somebody speaks a language that's close to the language, if somebody's speaking Italian and Spanish, maybe they'll understand each other better than somebody, speaking
Sure. Yeah.
Some other languages. So I think that's true in terms of people and their interpersonal interactions as well. So finding those compatibilities is just a matter of spending some time together and talking about the science.
I don't know if you want to add anything to...
And finding their strengths too, which is something I learned, not by being clever, but somebody once said to me that Shirley Tillman, who's now the president of Princeton, but she's a very accomplished molecular geneticist, and somebody who knew her very well in her science days said she always is very good at finding what people are good at and then making sure that gets used very well, and it's not an altruistic thing necessarily. She's making sure that they thrive and do the best science. And I thought that was a really good hint, and I try and look for that as well, because some people have real strengths in some areas. Some will ask all these questions all the time and they'll never do the experiment, but it's also important that they're doing that. Others will say, yes, I'll do the experiment, but also be critical. Carol was actually somebody in the latter. She said, I'll do the experiments and be very smart and critical at the same time. But other people will question. And if you can make use of that and say, this is really good, that someone's bringing to bear really critical thinking and not say, well, I really want you to do it this way. You stop and you think, this person actually is smart and they've probably got some good reasons for what they're thinking about. And so you try and use what strengths you feel you discern in people is important.
I like to find people who are pretty independent and have some initiative, and the best students are the ones where I can tell them that's never going to work, and then they go and do it, and I show that it does work.
My problem is I always think it's going to work, and they're the ones who say, well, actually, whoops.
It goes both ways. Yeah.
It goes both ways, yeah. And you have to have both going on. You have to have the sort of let's try it, and then things that you really have reasoned through very well. That sometimes that's the route to doing something new, is you reason something very well and you do that, and then something new comes of that too. So both ways in biology really can work, and you can't always predict which is going to be the formula.
Presumably it often takes quite a long time to find out what people are good at, because there must be a lot of graduate students who start and then find that they don't hit the ground running. It takes time to get going, and that can be quite a disillusioning time. So it's important for people to understand that it can take time for one to work out what one should be doing.
I think people's quality of thinking, that emerges relatively easily, I think. Now, as you say, what unfolds in the experiments can, of course, be very slow because, by definition, you're doing things that are difficult. If they were easy, someone would have done them. And so that's, I think, the very, the hard, unpredictable road for graduate students. And back to your question of mentoring, that's where you have to realize that that's going on and that people will go through periods in science, as we've all done, where, you're just seeming to fail all the time. And the experiments don't work, sometimes for reasons that are boring, but sometimes for reasons that are significant. And, you have to be able to fail a lot of the time. And scientists have to be terrific at getting slapped back on the face by nature all the time.
I've told my students, and I used to be famous in the lab for saying, that's why they call it research.
Yes.
Because somebody would come into my office and we'd be talking about an experiment, and there'd be a great result. I'd say, great, go do it again. Somebody would come into my office and nothing worked at all. Great, go try it again. And I said, that's why they call it research. You always have to do it again.
The opposite. Somebody said, is it research? Because somebody already searched for it and then you're just rediscovering it or something. I like yours better.
You have to repeat that good experiment.
I like yours, but
you have to try again at the failed experiment.
I like yours better.
Yes, Carol's is less depressing. Yes. So back to the experiment. Together you demonstrated that telomeres from one species could protect DNA from an entirely unrelated species, and thus the mechanism of telomere protection was more fundamental than perhaps one might have thought initially. And that result was very clear, and you understood that immediately. So were you aware of just what an important piece of information you just discovered at the time?
Well, I think we knew that it was going to open up a lot of new experiments, right? Because we could use all the tools available in yeast as well as what you could do in tetrahymena and then in other organisms. So we knew it was going to allow more progress.
And I think it also felt somewhat fundamental in the sense that, molecular biology was very dominated by there would be universal solutions for things, because I think we were so influenced by the genetic code and DNA, and everything was very universal. And so when you saw something crossing lines of phylogenetic divisions, I think, didn't you think there was a bit of a sense, and we found, the sequences looked similar and they looked like, there's something fairly deeply
universal
in the eukaryotic world.
Right. I think that's an important point, because it was already clear that in bacteria and many viruses there were lots of different solutions, so it didn't have to turn out to be universal.
Yes, yeah, it was completely non-intuitive what would what would go on in terms of actually more the replication problem, right? In terms of, well, and protection too.
Yeah.
For
Sure.
and yeah. Yeah.
And you also observed that the telomeres in yeast were lengthening and that something had to be causing that lengthening.
Yes.
And that's when you come into the story, because you set out to find the activity that was causing the lengthening.
Biochemically speaking, yes, because I thought that was, I'm sort of a biochemist, somewhat by training, I suppose, and I'd gone through biochemistry and then molecular biology, and so it felt natural to try and say, well, this is very direct. You know, reactions take place in real time, sort of more or less in front of your eyes, so to, you know, in a biochemical way.
But you had
Yes.
the right organism for doing the
And the
biochemistry.
organism was
And-
was right, and it turned out there was a biology of the organism that said there's a burst of telomere synthesis that takes place, and there was an abundance, relatively speaking, of telomeres, and so that all pointed to, well, you know, this is a good system to try and answer questions. And I'd been trained in the lab of Joe Gall, which is where I actually did the sequencing of the telomeric ends, as we now call them, but the DNA ends of the mini chromosomes in this ciliated protozoan. And Joe had very much always said, you know, you find the system in which you answer the question best, with, I think, this fundamental idea that things will be pretty conserved throughout much of life. And so this idea that find the good system was very much in my mind. So I started, you know, doing a little sort of foray into things, got tenure, felt brave, and Carol joined the lab and felt really brave.
So she was one of the unusual graduate students who took one of your ideas and said, yes, I can do that, and off you went.
I'd actually offered it to a postdoc who turned it down. Very nicely said, Liz, I think I'll do something different.
Okay, well,
Very politely, but...
So we might ask the name of the person who could be sitting there.
So that's very important that Carol, you know, was really not only willing but able, which is even more important in science, right? Anyone could be willing, but to be able as well is huge.
And you got this now famous Christmas present in 1984 of the first indication that you got your hands on the activity that was causing the lengthening of the telomeres.
Yeah, I was doing experiments. It was about 9 months of trying various things, right? Liz and Jack had proposed that there may be something which would lengthen the telomeres, and so not knowing exactly what that is or what the properties were, you know, we would just try different things, and Liz and I would talk to each other every day or so and say, okay, let's try this. I liken it to, you know, cooking. You add some ingredients and you taste it, and that doesn't taste so good. So you add a little bit more salt. So after trying various things, there was one particular change that I made in the experiment, and I was just interested in. It was an exciting time to, you know, do an experiment, and then a few days later, it would take several days for the experiment had to sit on an autoradiograph, and so I went back in on Christmas Day to develop the results of the experiment that I had done several days before, and that's when I saw this very clear repeating pattern on the autoradiograph that just looked like a 6-base repeating pattern that you would expect of a telomere repeat. And so that first instance, it's like, wow, this might really be what we think. Of course, then after the excitement, there's the, well, are we being fooled?
Oh, yes.
And so then has to follow all of the ways where we would then, you know, be our own worst critics in a sense. Yeah. It's like, how could we be being fooled by this? Maybe it's really some normal polymerase that's copying something that's a repetitive sequence in the extract. And so that's where the real work of the self-criticism is very important. And so that's why the discovery was Christmas '84, but the paper was published in December '85.
It's still pretty fast as these things go, actually. Right.
It's an obvious point, but obviously you were enjoying yourself tremendously, and going in on Christmas morning was just something that was natural. And the enjoyment of what you do is key, is absolutely the essence of it. It's not work, it's enjoyment,
Yes,
I
that's
presume.
right. It's the best kept secret in science. We never tell people what fun we're having, and maybe we're a little afraid because somehow society will frown upon the idea that you actually really, that's right, and yet it's serious play, you know, but it's completely the element. And there is always this, I wonder what because often the trajectory of experiments is that you find out something the next day because something or other has been incubating or autoradiograms exposing, things like that. So there's often this thing where you would leave something and then the next day come in. And I remember just driving to Berkeley, and I'd be driving up University Avenue, really impatient because the traffic would get very slow off the freeway, and you knew there would be something at the end of that University Avenue when you got out the car and went into the lab. And
That's why I lived up on the hill and I came
yes,
in
so
on my
you
bike.
came down and back fast. That's right. There you go. Right. Although your Volkswagen once,
Yes.
once she was nearly killed by her Volkswagen. At least you were on the bike. That was good. But I think that's really an important point, because many young people are saying, oh, science is so hard. It's so true, and we all complain bitterly because we just take completely for granted the fact that we are having such a good time. So we sort of have the luxury to complain about the other stuff. But it's a really good career and very autonomous. Nobody tells us what to do in terms of our choice of research. And when you think about how many jobs that's true for, it's immense. And it's not as if scientists waste money. I mean, we really are so driven. We want to find things.
I remember when I was a graduate student, and it was the first time that I was sort of on my own and supporting myself, and it's like, wow, they're going to pay me to come in and play every day.
That's right. Yes.
And I was being paid now what it would now be, but it was great. And I thought, wow, this is just amazing, and maybe if I just keep it up, and it's worked so far.
Yes. No, it's true. But as an assistant professor, you're suddenly given this playground, and they really trust me to do this. That was my feeling that, and Berkeley was very sink or swim actually, and so really they were just sort of entrusting you to go out, and I think now mentorship in young people's careers is much more thoughtfully done, and maybe that's not always good, because we had huge freedom just as an assistant professor, right? And you did things. Yes. I mean, you had to work hard to garner the resources, but then you worked really hard because you're just driven.
Perhaps it's too big a question to ask whether it's changing for the good or for the bad. It's presumably going in both directions at the same time.
I think those who love science are still driven in the same way.
Mm-hmm. Yeah. I don't know. I think it might be. It probably takes more time and effort to raise the money to support a lab. There's more frustrations there. Maybe it's more bureaucratic than it used to be. But if you really want to do it, you can still do it. And then do you have the luxury of doing whatever experiments you think are the most interesting?
Mm-hmm. Okay, back to the enzyme. The enzyme you discovered was unusual. It was a reverse transcriptase, a mixture of protein and RNA, and it took some time to sort all that out. When you did sort it out, you solved. It turned out that it solved the end replication problem, which is this problem that had lain around for a while unsolved, of how DNA polymerase. Well, the fact that DNA polymerase could not, on its own, synthesize both strands of DNA to the end. And it seems strange that problem had been there without anybody being able to solve it for quite a long time, because DNA polymerase was revealed in 1958 by Kornberg, and it was pointed out in 1970 or so, 1972 by Watson and others, that there was this problem. And then it was the late 80s, or the mid to late 80s, that the solution came.
Well, there were solutions in other organisms, as Jack had alluded to, that various viruses did it in certain ways. What wasn't known was how the eukaryotic chromosomes solved the problem, and that needed to wait for the sequences. And until Liz found the sequences, you couldn't really ask the question until you had the actual molecular details.
And there were some peculiarities about the sequences. There were different numbers of repeats at the ends of different molecules in a population of molecules. This wasn't like viruses, although there are some viruses in which they do have recombination at the ends and they have some end repeats as well. So there were solutions that looked very plausible that the viruses of bacteria had already, evolved to have. And so it was really various lines of evidence that sort of said, aha, this doesn't seem to be the kind of solution that eukaryotes have.
But if you go back and read all our early papers, right, there were a lot of models that were mostly based on recombination-like mechanisms. And we held on to those till the bitter end, until the sequences said, that can't possibly be.
Because people could draw beautiful diagrams, right?
We
I mean, the literature was full. You did, right? I don't know how to draw beautiful ones, so I was. No, but there was definitely some ideas out there, and some of them have turned out to be quite applicable actually to some viruses and stuff like that. So the ideas were not, completely useless.
Applicable
but
to when telomerase isn't there as well.
Yes.
Right.
So
Yes.
there really were
Interesting
two
aspect of it.
answers. It's just that
Yes.
one turned out to be a little bit more dominant than the other.
As in a lot more dominant.
A lot more dominant, yes.
Right, as in almost universal eukaryotes. But with exceptions, and exceptions are always instructive. And so, that's sort of the richness of the question, because there's such a lot that goes on into what seems very simple, protecting the end of a chromosome. Well, it sounds like you just stick a fortress around it and that's okay. But it's much more interesting and sort of dynamic than one had thought, which again makes it fascinating.
It is fascinating. Is it interesting because one tends to tell the story of science in retrospect as being a problem followed by a solution. And it's not quite that, is it? It's much more complicated.
And often observations come and then they aren't what you expect. And then you start thinking about them and what they might mean, and then realize, yes, that might be providing a solution to something. It doesn't happen in...
But there are also possible solutions that aren't correct, right? People pursue things and then they find out, oh, that's not how it works. And then you go back and pursue a different angle.
Yes.
And what you end up hearing about historically is those things that were correct. So it seems like a linear path when actually it was a branching tree, some of which were interesting ideas. They just didn't turn out to be how it actually worked.
Yes, or at least not in that situation.
And that, yes, how that particular thing.
Yes, yes. But I think...
And also, you get a solution to, say, the problem you were thinking about originally. But once you have that solution, you realize, oh, now there's 3 other problems we hadn't even thought of before. And so it goes.
Right, yes.
And that's really true in the case of like human telomeres, where the biochemistry is unbelievably complicated.
And yet, there's an inevitability to the complexity, because people say, well, if you want a very robust homeostatic sort of system, it has to be inherently complex. And so suddenly it sort of makes sense in a way that we didn't think of. We sort of really simplified the question, right? We just said, we're just going to think about the DNA and the enzyme that does it, and just, so you have to do that. I think up to a point, you sort of have to take away the irrelevancy.
As much as possible.
Right, and sort of, but always knowing that it's taking place in a cell, the incredibly complex entity which is a cell inside an incredibly complex entity which is, in our case, a human. And yet you can take these things and sort of push them up to a point. And then you have to realize when you have to take the blinders off. That's the key, right? but again, the inherent complexity of it is not. It's not like a curse. It's sort of like, this is the way it is, because there are really interesting reasons why I think systems have to be that way. And yet we have to, on the other hand, sort of reject the complexity at certain stages in the research.
It's a nice path of sort of going in and out of complexity and simplicity.
Right.
So, 20 years on, you're still working on telomeres and telomerase and sorting out the system. And one thing that's appeared... I'm coming to Jack. Coming back to that. One aspect that's grown up is the therapeutic implications, or the fact that telomere shortening was seen to be associated with disease states, and indeed the maintenance of telomere length in both directions seems to be important. Maintaining equilibrium is important. What do you feel are the potential therapeutic benefits of studying telomerase and telomeres?
Well, it's usually divided into 2 general categories. One is the rather hyperactive telomerase that characterizes the great majority of human cancers. And on the other hand, the telomerase that's present in much more regulated form in the natural cells of the body. The telomerase present in much more regulated form in normal cells of the body, and various indications such as the associations you mentioned of short telomeres with many disease states or risks of it. That's intriguing. There's genetic data in cases of insufficient telomerase action, which says that clearly is not good for humans to not have enough telomerase in their normal cells that have to replenish through decades of adult life. If we're fortunate enough to live, past our reproductive years and, we're looking at old age, we're interested in what our health is like for that. So understanding what's going on in cells at this fundamental level, I think you really do want to understand this in order to just understand what's going on in the trajectory of humans as we age, because all of the social and other settings are now letting us age. We have this sort of unexpected biological reality to live with. So we're understanding that, first of all. Now, are there quick fixes, magic pills? Not tomorrow, but maybe there's interesting things that could keep telomerase a little more active, but you have to be careful. And I like to sort of think it's like aspirin. Take 2 aspirins, good. Take a bottle, that's bad. Clearly any measure would have to be carefully thought through how you would, if you want to keep telomere maintenance better, not to push it too far, because too much telomerase can help cancer cells. But I mean really a lot too much. And then the question, of course, is can you exploit the high telomerase in cancer cells to selectively target cancer cells? And there's beginning efforts in industry to do very early stages to look at these kinds of things. So, but I think just understanding what's going on is actually really important in understanding human aging.
And it's not just telomerase, because one of the things that we've learned in the research over the years is that it's really the short telomeres that cause the end effect. As Jack had mentioned, anytime you ask a question and you find out the answer, there's many more other questions to ask. And so there are various regulatory mechanisms that allow the telomere length to be maintained at a certain equilibrium. And telomerase is essential to provide the raw material to do that. But both the telomerase is regulated as well as the proteins that are on the telomeres that the telomerase has to interact with. So I think that really understanding those details of all of the components and the complexity that goes into the regulation will tell us a lot about these diseases, these age-related degenerative diseases that may not be just telomerase related, but there may be a number of other genes that one could look at that may be associated with these degenerative diseases that aren't directly the telomerase. So there's a lot of interesting avenues to pursue still to really understand the different directions these diseases may come from.
Because there was a great deal of therapeutic excitement about telomerase and telomeres early on, and there still is. But was there an initial kind of pressure on you suddenly that everybody was getting excited about the potential? Does it make life difficult if people's expectations are a little bit too elevated early on?
Well, I work in the basic science area, and so I felt immune from that.
It's all the companies that have to worry about that kind of expectation, because we never really said it as scientists that there was going to be any kind of therapeutic coming tomorrow, because we didn't really have the vested interest to be doing that.
Yes. No, I think it's good to have these avenues explored for sure, and I think that the fact that it hasn't gone all that fast is actually not to do with the science, but other sorts of things. And in the meantime, it's really important to try and understand what is going on, because any therapeutic is going to be all the better for having a better sense of what underlies its usefulness and where its danger points might be. And it's not clear, how we think about these issues of long-term diseases that affect huge numbers of people. You don't necessarily put everybody on statins. I mean, that's a common thing. But perhaps that equivalent isn't necessarily the best way to go either, although we tend to be a very take-a-pill-oriented society. And there's nothing wrong with that. I mean, I'm all for Western medicine, believe me. But I'm just saying that we don't want to limit our thinking, I think, to that.
Jump into something too quickly.
Or jump into something.
Recognize a problem.
Exactly. That's right. Right.
So I wanted to end just by dealing with this question of staying with your subject or not. So you two have stayed with the subject.
Well, let's debatable.
Okay.
The subject keeps changing.
That's,
The subject really has changed continually.
I'm sorry, and the subject expands.
Yeah,
So that
yeah.
it's,
I was a biochemist, and now I'm working on recombination and human disease and various other things.
Right.
So.
I work with clinicians on chronic psychological stress. I mean, but the point is, I'm not the expert. I bring my expertise and they bring theirs.
yes.
So it stays very fresh by keeping one's expertise that you really have, now interfacing with other expertises. So it's actually a very broad topic. Anything that says our cell's going to be able to keep replenishing, that's got a lot of broad implications, even though we're focused on one part of it.
Well, maybe I'll discover my question is completely redundant. But let's say you two are at least following following the questioning in the same general vein, and yes, it's taking you to new places. You, on the other hand, have seemed to jump from one question to another. There's almost a clear break between one line of questioning and the next, and they seem from the outside to be two different ways to do science. One is to say, there's a problem, I'll work on it for a while, and then I'll look for another problem, actively go and find a different problem. Would that be fair to say? I don't know, initially.
I think you could find lots of examples of people who, have one system and they use it to address lots of different questions. And, that can be extremely productive. And then there are other people who just like to find interesting questions in different areas and go for it.
But what I was going to ask was what for you is the attraction of jumping from question to question?
Well, it's fun to think about new things, get into an area where you don't really know very much, so you're not being fooled by the preconceptions that might dominate the field. So you might have a chance of making a contribution in a different way. So that's part of the attraction, I think.
Is there also an attraction about going to less populated places?
Yeah, for me, I don't like to be working in an area where there are a lot of other people who are going to do the same experiment, at the same time or a few months later. I find it more fun to be doing something that is probably unique.
And that's what kids were initially too.
Yeah,
nobody else was doing.
Nobody
was asking these questions.
Right.
Yeah.
Yes.
I think it is the most fun way to do science, actually. I agree with you.
Right. So in the mid or late 80s, I think the implications of all the telomere work were becoming clear, and it was, I think, clear that a lot of people would be going into that field, and so I think that helped to make me look around for other areas. And all the stuff about ribozymes was very new and exciting. I was very surprised that there were very few people going into that area. So I thought,
It had already
we
won the
might.
Nobel Prize by that time.
Well, that was '89,
okay.
and we started working on it in actually '85.
Yes.
Yeah, so it was fun.
And how life begins. I mean, that's a pretty important question, right?
That's a lot of questions, actually,
Yes.
when you start to
Yes.
break it into pieces.
component
It's a lot
parts,
of
yes.
interesting questions. Right? So
Yes.
that's come to
Yes.
dominate what we do today.
On the note that there are lots of interesting questions left, may I thank you all very much indeed for this talk and wish you a wonderful Nobel week in Stockholm.
Thank you so much.
Thank you.
Thank you very much.